Good afternoon. Welcome back for the last time to virology. But this is not the last time you will encounter viruses. Don't forget this, okay? It's very important stuff. Today, this is like the ultimate lecture. You've learned everything you need to now understand what's going on. You know, you opened your there.
It's a beer.
That's not a beer, it's water. Once I was doing a podcast in front of like 500 people, and they're opening their cans. So I said, just open them all at once, please. Let's get it over with, and they did. That was pretty cool. We're going to build viruses to help people. I've been telling you all along about how viruses are either neutral or harmful. Today, we're going to harness them to help people. And we call this therapeutic viruses because we're going to do a variety of different things. Now, I'm going to leave you with that last quote. My colleague, Dixon Despommier, who podcasted with me for many years, unfortunately died in February. He used to say, trust science, not scientists. And that's so true, because individual scientists can be wrong, but the whole field will fix it. If it's important, if someone makes an observation that's wrong and it matters, someone else will fix it. It doesn't always matter what people discover, but if it's important, someone will fix it. So we're going to talk about 4 different ways to use therapeutic viruses. First, we're going to talk about using phage therapy for bacterial infections. We're going to talk about gene therapy. Many people are born every day that lack a gene, and therefore they have a disease of some sort related to that. So we're going to talk about using viruses to deliver that gene to them. We're going to talk about using viruses to deliver antigens as vectors for viral vaccines. And then finally, we'll talk about viral oncotherapy, using viruses to cure cancers. All very exciting stuff. This is a big future in virology. If you're interested in the field, these areas are, in my opinion, very interesting to do. A key for the development of everything we're going to talk about, we're going to all talk about modifying viruses. Okay, and to do that, you need to modify a DNA copy of the genome, which here is shown for poliovirus. So poliovirus is an RNA virus. Its RNA, when extracted from the virus particle and placed into cells, is infectious. Because it's a plus-stranded RNA, it can be translated, and all the proteins are made to initiate a replication cycle. You can take that viral RNA and make a double-stranded DNA copy of it in a plasmid, a bacterial plasmid, and that plasmid can be propagated in bacteria. You can introduce that DNA into cells, and the DNA will be transcribed, and you can initiate an infection. And this is very cool because DNA can be modified now. You can put genes in, you can take genes out, you can alter genes. So all the things we're going to talk about today is made possible by this observation. And more recently, we can make in vitro transcripts of this DNA and put those in cells, and those are infectious. So this is an experiment I did as a postdoc in 1981 with poliovirus. It was the first time anybody had made an infectious DNA of an animal virus. So I've been manipulating viruses for over 40 years. I have a lot of experience with it. When I hear people say, I don't know if you heard this, but some guy blamed the measles outbreak in Texas on a bioengineered measles virus that was made more pathogenic. Did you hear this? This is nonsense. Do you know how hard it is to make a virus more pathogenic? We have no idea how to do that. If they think, oh, you just go in the lab and you put a few bases in and you make it more pathogenic. No, we have no idea. And I should know, I've been doing this for so many years. But some lawyer somewhere just says they made it more pathogenic. Okay, so be wary of these statements. You're not going to have me anymore each week. So think on your own. I've given you the tools to think. And if you don't figure it out, you can always contact me. All right, so that's a key discovery. I'm not bragging. I'm just saying it was pretty cool that I did something that mattered, because we don't all do, you know? Okay, phage therapy. So in 1915, this guy, Derel, discovered bacteriophages. He found that these phages, these viruses of bacteria, kill bacteria on a plate. So he thought maybe we could use these to treat bacterial infections. Remember, there are no antibiotics at this time in 1915. So he co-founded the Eliava Institute in 1923 in Tbilisi, Georgia, not the US Georgia, the one in Asia. And they pursue phage therapy. In fact, during World War II, soldiers in certain armies were given kits with tubes of phage in them in case they got gastrointestinal infections. Now, unfortunately, in the 1930s, the antibiotics were discovered and people forgot about phage therapy because the antibiotics were cheap and could be easily used. But the problem is now we have widespread emergence of resistance to antibiotics, right? Almost many bacteria are resistant. Tuberculosis is completely resistant to every antibiotic we have. Certain strains of tuberculosis and many others are becoming more resistant. So you're going to see more and more problems with bacterial infections. So people have gotten more interested in phage therapy. That is, you have a bacteriophage that kills bacteria, so you treat patients with that. It's brilliant, right? It's very simple, makes sense. So the idea is you use lytic bacteriophages. You know what that word means. You should know what it means. To kill a specific bacterial host. And you don't want to kill your flora that are beneficial, right? So you can't have a phage that kills everything. So if you have a particular infection, what they will do is isolate phages specific for that bacterium. And the idea would be in the future to have a whole collection of phages that you could have on a shelf, and then you could screen them for the ability to kill your pathogen. So that's why I have bullet point 2. Pathogenic bacteria must be identified, and the phage sensitivities need to be determined before treatment. So this is not so simple. Plus, just think about FDA approval. How are you going to approve a drug that's tailored for each person? So this is going to require some tinkering of the regulations. But in the meantime, some other things have happened that are interesting. First, phage license. Phages that lyse hosts do so with one or more proteins that they make. These are called lysins. You can purify them, and if you add them to surfaces, for example, they will lyse the bacteria on the surface. And so some of these lysins have been approved for surface contamination of, say, food areas, food preparation areas. Agriphage is approved by the EPA in 2005. RIP EPA, that means rest in peace, because they want to get rid of it. Approved by the EPA in 2005 to treat a tomato disease caused by bacteria. And then List Shield is a listeriaphage. So listeria is a bacterium that commonly contaminates food products. So, you know, you go get your liverwurst and it's got listeria in it. Who knows why? Maybe it's on the slicing machine, came from somewhere else, but it can cause serious infections in people. And so this phage was approved by the FDA, not to treat people, but to treat contaminated meat and poultry. And that was the first phage to get this designation of GRAS. Do you know what GRAS is? It's not fat in French. No? God, you don't know? I'm not making fun of you. I'm just surprised you don't know. Generally recognized as safe. I guess there are too many acronyms in the world, right? Okay. Generally recognized as safe. That's a designation that a product has to get before it can be sold. The idea is to use phage therapy to treat human infections. And so obviously people have tried this experimentally. And these are just a couple of examples. They've been published. The one on the upper left, phage treatment of an aortic graft infected with Pseudomonas. So a patient got an aortic graft. It became infected with Pseudomonas. It was resistant to antibiotics. And so they decided to treat it with a phage. So the surgeons opened the patient up and they squirted the phage onto the graft. They got rid of the patient's Pseudomonas infection. Unfortunately, the patient died on the day the paper was published. But it worked for him. It's not funny, right? But it's ironic that he would die on the day of publications. Of complications, not with infection, but just his heart was problematic because he had an aortic graft, so not so good. That one on the bottom is very interesting. Paul Turner, by the way, on that top paper is at Yale, and he was our guest on the podcast Twevo 44. Talked all about this. And then at the bottom, we have engineered bacteriophages for treatment of a patient with disseminated drug-resistant Mycobacterium abscessus. So this was an immunosuppressed patient that had M. abscessus that could not be cured with antibiotics. And so they engineered phages that were specific with this. Now, these phages are very interesting because there's a high school program throughout the world called C-phages, which was started by Graham Hatfull. This guy, he's at the University of Pittsburgh. He has high school students in the guise of biology classes. They go out in their backyards and they get dirt and they isolate phages from the dirt. And they're usually mycobacteria of different kinds. They get to sequence the genome. They get to give it a name. They have all sorts of creative names. And anyway, one of those phages was used in this patient because it was a match for the strain. And the interesting thing about this is that as treatment went on, I believe the mycobacteria developed resistance to the phage treatment, so they re-engineered the phage to overcome that and treated him with that. So it's amazing the things that you can do. And then finally, this one on the right, this is a very famous case, development and use of personalized phage-based therapeutic cocktails to treat a patient with disseminated resistant Acinetobacter baumannii infection. So this was a patient who was I think he was in Turkey for Thanksgiving with his wife, and he got very sick, and they couldn't fix him in Turkey, so they airlifted him to Frankfurt, and there in the hospital, they said, you have Acinetobacter, which is a very severe bacterium, and he said, we can't treat it. It's resistant to everything. So he went into a coma, and his wife is here on the paper, Stephanie Strathdy. She actually wrote a book about this. What's the name of the book? The Perfect. She worked at the UCSD, University of California, San Diego, and for one day she Googled phage therapy and she came up with all this stuff. So she called his doctor and said, do you think we can try this? He said, why not? He's going to die. So they isolated phages and they started giving it to him intravenously, like 10 to the 12 phages a day. And he's alive now. He was in a coma, and after 14 days, he woke up. IV. So the people who made the phage, we interviewed on that episode of TWIV. But then I interviewed Stephanie Strathdy last year at South by Southwest. And if you go to South by Southwest and search for phage therapy, you can find it. Very interesting interview. Anyway, so these things sporadically work, but there's no approved therapy because there's still a lot of problems. Here's one of them, that you get antibodies made against the phage, especially if you introduce them intravenously, and those antibodies can interfere with treatment. So that's something that has to be dealt with. Okay, so that's phage therapy. So it's just a little taste because you get an idea of the approach, but there are lots of difficulties, so we're not there yet. We're close, but I think for certain applications, eventually it will be used. Now, for the rest of the talk today, we're going to deal with mammalian hosts, not bacteria. And so we use a set of vectors, viral vectors, to deliver genes, to make vaccines, to do oncotherapy. And I want to talk about the vectors that we use. And there's specific reasons why we use each of these vectors. First, adenovirus, which is the DNA, double-stranded DNA virus that we talked about a lot with about 34 kb double-stranded DNA genome. And all of the research that we have done on adenovirus allows us to know how to modify this virus so it doesn't kill you, and so you can put genes in it. So these are very useful for gene therapy because they efficiently infect post-mitotic cell. What is a post-mitotic cell? It's a cell that doesn't do mitosis anymore, like many cells in our body, neurons, etc. Fast gene expression, it's episomal, so it doesn't insert. You can put up to 37 kb in it. It's easy to make pure preps. And there are a lot of serotypes, both human and animal, so you can give different serotypes if you have immunity to the vector. But the viruses can cause acute infection in people, so you have to get around that somehow. And so a series of generations of vectors have been made. The first is first generation, where we have deleted E1 and E3. And so E1 encodes the T antigens. Now remember, E1 products kick the cell into division. So without E1, these vectors are not going to replicate. They're going to deliver the gene, and that's it. They're not going to make new particles. And E3 is not essential. That contains immunomodulatory proteins. So these two deletions give you some space to put foreign genes in, but it's not a huge amount of space. Second generation, they added deletions in E2 and E4. So we have E1, E2, E3, and E4. So you got more space for the transgene. And then perhaps the most interesting are third generation, which we call gutless vectors, because there's nothing in them but the ends. All the genes are gone, right? The only thing you have left are the two inverted terminal repeats and the packaging sequences, about 500 bases of packaging sequences. This is the vector. Here's the whole genome as a comparison. So just the two ends are basically here. And then you put your transgene in at the right end, downstream of a promoter, okay? And this, of course, requires a helper virus because it can't package on its own because there are no capsid genes. There are no viral genes whatsoever. So how you use the helper is amazing. This is the stuff you can do with cloning. So the helper virus, so here's our vector right here on the top. This is the helper virus at the bottom here. So it's a full-length adenovirus which has two lock sites flanking the packaging sequence. So on the left is the packaging sequence. Here we expand it here. And these are the two lock sites. LOX is a target for an endonuclease called CRE. So it's the CRE-LOX system. And you can remember that by cream cheese and lox. That's why they named it, actually. So you put the lock sites in here on either side of the packaging sequences. And so it's also deleted for E1. So you take this DNA and you put that into cells that produce CRE and E1. So this DNA can replicate. And you also put your vector in the same cell. So the helper produces capsid proteins, right? So those are late proteins. Remember, you need to have this DNA replicate to get late proteins. That's why you need to put E1 in here. But CRE is being produced in these cells, so it cuts out the packaging sequences of the helper, so the helper itself will never be packaged. Because you don't want helper virus in the patient. That's bad. That will replicate and cause problems. So this allows you to make vector preps that are free of helper. So this is really, really clever kind of thing that you can do. Only the vector DNA is packaged, okay? I'm going to give you some examples of the use of these in a bit. Now, a very powerful and useful vector is adenovirus-associated virus, okay? These are the single-stranded DNA viruses within icosahedral capsids that we have talked about And you probably said, why do I have to learn any of this? Well, here it is. It's an amazing vector for gene therapy. Single-stranded DNA that encodes only a couple of proteins, the rep involved in DNA replication and the capsid, encapsidation. And so you can delete most of those. So here's the genome of wild-type AAV, the rep and the cap genes, 2 ITRs. You delete them, so you can put a transgene in. You leave a promoter and the 2 ITRs, and then you transfect those into cells. Now remember, AAV needs a helper virus, which is adenovirus. Well, we don't want to put the whole adenovirus in here, so we have identified the add helper functions that are needed to have AAV replicate, and those include E1A, E1B, E2, E4, and the VA genes. Okay, so you put those in on plasmids, and then you also put a rep cap plasmid for AAV so it can be packaged. And this now has the vector with its transgene in it, and it's packaged in an AAV capsid and nothing else. And the cool thing about this is that you get long-term gene expression without integration of the DNA into the host cell, because as you will see, integration is bad. Now, what's very interesting is that AAV can integrate into the host genome without a helper. But that requires the rep proteins, and those are not made in vector-infected cells. So there's no integration. Rather, what happens is the vectors recombine and make circular monomers that persist for a very, very long time, years, as far as we can tell. And in many cases, that can be useful. The quizzes are back, if only for one session. First question is, which technology is indispensable for the production of therapeutic viruses? X-ray crystallography, high-throughput genome sequencing, synthesis of infectious DNA copies of viral genomes, plaque assay, immunofluorescence. Which one is indispensable? Only one. So pick the best choice. All right, that's pretty good. Let's see how we did. So C is the right answer. It's the best. I mean, you need to do plaque assays, probably need to do sequences, but you can't do any of this without synthesis of infectious DNA copies of viral genomes. Retroviruses are used as vectors also. Here, the strategy is very interesting. You have 2 plasmids. You have a capsid plasmid, which encodes the structural proteins GAG-PAL, and then you have an envelope plasmid, which encodes the envelope glycoprotein. When you put these 2 plasmids into cells, you actually make virus particles, but there's no genome in them because you haven't put a genome in. But it's interesting that all these proteins can direct the synthesis of capsids with spike proteins. So if you want to introduce a transgene, you add a 3rd plasmid, the transgene plasmid, and you have a promoter driving your transgene, and you need a packaging sequence to get this RNA into the virus particle. And then you have a retrovirus containing your foreign gene. So that green is the packaging sequence. Remember, that allows the RNA to get packaged into the particles. Again, basic research unveiled the packaging sequence. We didn't know we were going to use it for construction of vectors. So most vectors are based on lentiviruses like HIV, which is amazing, right? We have a virus that has killed millions of people, and we can use it as a vector. We've made it safe and use it as a vector. And HIV is good because it can infect non-dividing cells. Again, lots of cells that you want to put genes into are non-dividing. You get long-term expression because the genome is integrated as a provirus into the cell. You can get up to 8 kb inserts, which is good. However, remember, there are 2 LTRs on the left and the right. And so when the genome integrates, and becomes a provirus, there's the possibility that the right-hand LTR promoter will transcribe a gene that you don't want to be transcribed, like an oncogene, and that can be bad. So you have to modify the LTR. Now, HIV, bona fide HIV only infects CD4 cells, right? But you can put a different glycoprotein in to give it a broader host range. You can put in, for example, the glycoprotein of VSV, and we call that pseudotyping, and now it will infect many different cell types. Or you can put whatever glycoprotein you want that targets to the specific cells you're interested in. We use Poxvirus as vectors. Poxvirus has killed many, many millions of people over the years. Again, we have modified it so it's not pathogenic. This one is, the vector is called modified vaccinia virus Ankara, which was originally made as an alternative smallpox vaccine, and it is currently part of the U.S. Strategic National Stockpile. Did you know we had a U.S. national stockpile? Yeah, we have vaccinia, we have smallpox vaccines, we have antivirals, we have other vaccines in case of an emergency. This virus has been designed to be replication deficient. It reproduces in avian cells, but not mammalian cells. And the reason was it was passaged extensively in avian cells, and it reproduces in them, but it's lost the ability to grow in mammalian cells due to an assembly block. The other nice thing is that you can work with this virus under BSL-1, the lowest biosafety level, right? This is your high school lab, is BSL-1, has large capacity. And there's also another poxvirus, canary poxvirus, that has been similarly modified. So the way you make recombinants is very interesting. Poxviruses are very recombinogenic. So you take the transgene in a shuttle vector. So basically, the blue is your transgene and the red are sequences homologous to the genome of the vector, in this case, Ankara. You infect with MVA and transfect this plasmid in. It's going to recombine into the viral genome because of these recombination sequences that target it to the viral genome. Very easy to do. Any other viruses can be used as vectors. VSV, remember, is a negative strand virus that makes 5 mRNAs. So what you can do is you can replace the glycoprotein gene here in red with a transgene. And the way you recover virus is you take 4 plasmids encoding the N, P, and L proteins. You have a plasmid with encoding the RNA genome with a transgene in it. Put those into cells, and then you infect with vaccinia virus encoding T7 RNA polymerase, because all of these plasmids, transcription is driven by T7 RNA polymerase. And out comes VSV with a different viral glycoprotein in it after this multiplasmid transfection. Flaviviruses, remember those? Dengue virus, yellow fever virus, West Nile virus. Positive strand viruses have also been used as vectors. For example, we have taken this is actually the yellow fever virus genome, and specifically it's the vaccine yellow fever virus genome. So we can replace these 2 proteins, M and E, with those of dengue virus and automatically get a dengue virus vaccine, which has been licensed in some countries. You simply take this RNA as a plasmid, you make your modifications to replace the structural proteins with dengue virus, make RNA transcripts, transfect them into cells, and out comes your new vaccine virus. And alpha viruses can also be used as vectors. These are plus-stranded viruses that are unique because they have a subgenomic messenger RNA. And you can replace the structural proteins with structural proteins from an alpha virus that is pathogenic for humans and make a vaccine that way. And finally, Newcastle disease virus is actually a virus of chickens. And here you can insert a transgene between the P and the M genes. You do the 4 plasmid transfection, and out comes Newcastle disease virus with an extra gene. And so this Newcastle disease virus is in itself used as a vaccine for chickens because it causes a serious chicken disease. And in one application, the H5 hemagglutinin, right, from high-path influenza virus has been inserted in here. So now you can immunize chickens against both H5 and Newcastle disease virus at once. So lots of different ways that you can use vectors. And these are some licensed vaccines that use viral vectors. There's one, the Zaire Ebola virus glycoprotein in a VSV vector. It's called Ervibo. This has been licensed in the US and it's used extensively in Africa when there are outbreaks. Denvaxia is the dengue vaccine made by exchanging the structural proteins of the yellow fever vaccine with dengue virus. The yellow fever vaccine vector has also been used to make vaccines for Japanese encephalitis virus for people and West Nile virus for horses. Now, in this country, we don't have JEV, but it's endemic in many Asian countries. And so if you're traveling there, you should be immunized against it. And West Nile for horses, of course, West Nile is very bad for horses. So if you have a $100,000 racehorse, you better immunize it. And as I said, the Newcastle disease vector for H5, there was an AD26 vector developed with the spike protein of SARS-CoV-2. It was licensed or given an emergency use authorization early on, but I don't know if you remember, but it caused severe blood clotting issues in a small number of patients, some of whom died, so that's no longer used here. There's another adenovirus vector, ChAdOx1, chimpanzee adenovirus. This is a chimpanzee isolate of adenovirus, which has been used throughout Europe to immunize against COVID. And this is nice because nobody has antibodies against chimpanzee adenovirus, so you don't need to worry about seropositivity. And finally, Sputnik is the COVID vaccine made in Russia. And they called it Sputnik because they were the first to make a COVID vaccine. And it uses two different vectors, AD5 and AD26. Okay, now an interesting technology which may or may not come forward is self-amplifying mRNA vaccines. So we've talked about mRNA vaccines where you take an mRNA, coat it in a lipid nanoparticle, you inject it into a patient, and the patient makes spike proteins. But self-amplifying mRNA vaccines have the ability to replicate themselves. They encode an RNA polymerase, so you amplify the mRNA and in theory, get more spike protein made. And so this is kind of a summary of these vaccines. So here is, these are two replicon RNAs based on either alpha virus or flavivirus genomes, which have mRNA synthesis in them. They will amplify themselves. So the gene of interest is brown here, and the blue are the viral sequences, mainly the self-amplifying genes. And so these will amplify, but they will not propagate. They will not make new virus particles. On the other hand, you can make recombinant viruses, as I've told you, with flaviviruses. Put your gene of interest in. These are self-amplifying, but the downside is they make particles, right? So maybe not a good idea to do that. I think the future of mRNA vaccines is in doubt. The current public health people don't seem to like them for reasons that are not clear because there's no evidence that they're bad. And so we may see them canceled. Who knows? Okay, so now let's talk about two applications of these vectors. First, gene therapy for monogenic diseases. So monogenic disease is caused by a mutation in one gene. There are over 6,000 different monogenic diseases, and the frequency is 1 out of 200 live births. So 1 out of 200 live births the child has a mutation in one of these 6,000 genes that predisposes to monogenic diseases. So because it's one gene, monogenic, this is amenable to gene therapy. You can replace one gene. And at the moment, there are over 1,800 clinical trials in progress to assess different treatment of different diseases, although some have been already licensed for treatment. So these are some of the diseases that are of interest. These are monogenic diseases, the defect, incidence, and the vector that's been used for the investigations. So SCID, for example, is a severe immunodeficiency, which is being treated with retrovirus vectors. Hemophilia and hemoglobinopathies, deficiencies in factor IX or globin genes are being treated. This one here, retinal degenerative disease, is being treated with AAV. And we'll talk a little bit more about those and others as well. But you can see reasonably high frequencies, and so amenable to producing vectors and testing them. The numbers of clinical trials using viral vectors for gene therapy has gone up in the past years, as you might imagine, as there have been more successes. So this is an old graph, but you can see a few hundred ongoing. And these work in two ways. One way, you can take your virus, you put your gene in the virus by the methods we've talked about, and you just inject those into the patient. So here, let's put in an IV drip, or you can use cell-based delivery. The first approach, all the virus will go to the liver and mostly be cleared. So if you're replacing a liver gene, that's fine, but most other therapies, not great. So what you want to do is a cell-based delivery system where there are two different approaches shown here. One of them is you take stem cells from the patient or from a stem cell bank or embryonic stem cells, and you infect those stem cells in culture outside of the patient with your vector, and then you put the stem cell, and you put the cells back in the patient, the stem cells, and they will differentiate into various cell types carrying the vector DNA. An approach we'll talk about later, you can also make what are called CAR T cells. You take T cells out of the patient and you put your vector in them, and you give the T cells the renewed ability to kill, say, cancer cells specifically. But we'll talk about that in a bit. So the earliest trials of using gene therapy to treat monogenic diseases were in the 90s. First one here, one of the first, a 23-year-old male with cystic fibrosis. So this disease, a lung disease, is caused by mutation in the CFTR, the cystic fibrosis transmembrane conductance regulator gene. So we know it's a specific genetic defect. And so this patient, who you can see lying there with his physicians all around him, is being given 10 to the 8th PFU of a E1, E3 deleted adenovirus with the CFTR DNA inserted into the vector. So they produced this virus in the lab, grew it up, and they basically sprayed it into his lung. They intubated him and sprayed it into the lung. Of course, the tube has a camera on it. You can see them looking at the TV screen. And this resulted in some expression of the transgene. So you can see they gave him one injection, and pre-therapy, these are some of the lung cells, and post-therapy, they stained with an antibody to the CFTR, and you can see they're making protein. And if you can quantify it here, you see there's a peak of expression that lasts maybe 30 days, and it goes down to zero, and they give him another one, another injection, another peak. But by the third one, he has so many antibodies, he doesn't make any more protein. All the vectors neutralized. So, and this patient didn't have any clinical recovery. It just is a proof of principle that you could make the gene, but it's not the right approach using an adenovirus vector. So many other gene therapy trials were initiated in the meantime, and then in 1999, we had the first death in a gene therapy trial. This was unfortunately this young man, Jesse Gelsinger, who was 18 years old at the time. He had a disease called ornithine transcarbamylase deficiency. It's an X-linked disease. You get a lot of pneumonia and glutamate in the blood. His was not so serious, but he and his dad decided he wanted to contribute, so he enrolled in this trial. But if you have severe form of disease, you have cognitive problems and premature death. So he and other patients were given an adenovirus vector with the OTC gene in it at UPenn. And unfortunately, Jesse died 4 days later from a massive immune response and multiple organ failure. And so now an examination of the data at Penn showed multiple rules of conduct broken. The trials throughout the US were all stopped. Certainly the ones at Penn were stopped. They did a lot of things wrong, which you can read about online. You can go find out. And his dad, in fact, has written extensively about this. So this was just an example of people too excited to do things without being safe enough. There had been warning signs that would lead to this, but they ignored it. Everything now is fixed, presumably, but this didn't need to happen. Now, another example of a disease that's treatable is X-linked severe combined immunodeficiency. These are people that lack T cells, B cells, and NK cells, so severely immunocompromised. And the defect is in the IL-2 receptor gamma chain. This is the gene for it here. And these colored symbols are mutations in a variety of people with X-linked SCID, this is called. And so this is amenable to gene therapy. And some years ago, 2 trials were initiated in London and Paris. They gave infants. So you have to give it at a young age because you're likely to get infected and die later on, right? Unless you live in a bubble. So they gave this vector containing the normal IL-2RG gene to infants. So they took out bone marrow hematopoietic precursors from the child. They put them in culture. They put the retrovirus vector in them. They confirmed that the protein was being made. Then they put them back into patients. And so these bone marrow cells making the normal IL-2RG are now populating the patient. And in fact, these children were cured of SCID. But years later, 4 out of 9 infants in Paris and 1 in London developed T-cell leukemia after treatment. Now remember, this is a retrovirus vector. And it's one of the earliest ones. So all these trials with retrovirus vectors were stopped, and this was studied extensively. And it turned out that the vector inserted next to oncogenes in those kids, right? Remember, you turn on an oncogene, cells start to divide, they accumulate mutations, and you have cancer. So this is a map of the insertion sites. So we have 2 different integrations. One here. This is the oncogene LMO2, and another one here. And these led to turning on of the oncogene, and that caused the leukemia. Now, these kids were treated, and their leukemias regressed. So that's the good news there. And also, vectors were redesigned so the 3' LTR wouldn't have an active promoter any longer. And so that solved this problem. But you can see, you have to take risks, otherwise you don't make any progress. So here's an example later. 8 infants with SCID got bone marrow transplants again with lentiviral IL-2 receptor gamma chain infected bone marrow stem cells, this time with modified vectors. And after 18 months, all the kids had functional B and T cells. Sickle cell disease is also treatable with viral vectors. So in sickle cell, of course, you have a mutated beta globin gene, and it's altered and causes a defect in the formation of our red blood cells. And so there are a couple of ways that you can fix this. One vector is you deliver the normal gene with CRISPR-Cas9. And this basically cuts the donor DNA and recombines into the cut site and corrects the defect. So you have DNA in there that is correct, and it will be replaced into the site with the mutation. And then on the right, you have lentiviruses that encode the correct transgene, and these are used to infect, I believe it's used to infect stem cells, and those are replaced into the patient, and you get healthy red blood cells. So these are now approved gene therapies. I think I have some prices later on for your interest. It's not cheap, folks. These are very cool, these inherited retinopathies. These are untreatable blinding conditions. They're caused by monogenic mutations in retinal photoreceptors and the retinal pigment epithelium. So, here's your eyeball, and at the back is the retina. The light shines on it, and there are receptors in there that transduce the light to your brain. And so this is what it looks like, right? Here's the retinal pigment epithelium, and they pick up the light. There are a bunch of genes in those that are defective in people who go blind. And then you have a series of cells beneath. Remember the cones and the rod cells from high school biology and others that eventually transduce the signal to the brain? And there are genes in each of these cells that are defective and lead to these blindness. So AAV, lentiviral, and adenovirus vectors have been tested to deliver these different genes in different animal models. And it turns out AAV is the most promising for this. And they test them in animal models first, and then in humans. And what they do is they take a needle, push it through your eyeball to the retina, and inject the AAV into the retinal tissue. It probably hurts, but you see again at the end. It's really, really amazing.
There's a documentary on Netflix about
Yeah.
a kid who...
Yeah, the kid got it, and he said, I saw my mother for the first time. Oh, how much is that worth? Is it worth $900,000 for both eyes? I guess. Anyway, this one, you can't ask me, I'm too old, right? I'm jaded. The Leber congenital amaurosis is a disease with specific mutations in this RPE65 gene, which is needed for photoreceptor function. And so they tested in dogs first a single subretinal injection of AAV with the canine gene. It restores visual function in genes, although the dogs can't say they saw their mother for the first time, but I guess you have other ways of assaying it. And then it was trialed in people, and it was a success. So FDA approved in December 2017. Luxturna, it's called. $895,000 for 2 eyes. You got to get your insurance company to pay for it, right? But I don't know if they will. Why is it so expensive? How much is it worth for you to see is what they'll tell you. Anyway, in 350, I interviewed Catherine High, who was a professor at Penn who ran these clinical trials, and she talked all about it. Very interesting stuff. I think this is a great success story. So the thing is, these AAV vectors persist, right, for years. And so you have your vision for years. Who knows how long, because the earliest ones aren't that long, but we'll see. It's very cool. All right, some other viral gene therapy successes here. SCID, I told you about. Cerebral adrenoleukodystrophy. They give the gene to people that's defective. It's called the ABCD1 gene. $3 million. Well, this is a very rare disease, so I don't know. I'm not going to talk about the economics, but there it is. Sickle cell disease. These are some approaches that I've told you about, either delivering globin, Lyfgenia, or Casgevy, which has the CRISPR-Cas edit. Beta thalassemia is corrected with a lentivirus. Hemophilia A and B, you deliver clotting factors 7 and 9 in AAV vectors. $3 million for that also. Lipoprotein lipase deficiency, a fat metabolism disorder. You deliver this LL gene, lipoprotein lipase gene, in an AAV vector, $1 million. Spinal muscular atrophy, you deliver the SMN1 gene in an AAV9 vector, $2.25 million. And then Duchenne muscular dystrophy, DMD gene in AAV. So I guess the most expensive are $3 million, but who knows? This probably went up. Hey, with tariffs, it's even going to be more, right? Who knows what components came from somewhere else? Second question. Which of these viral vectors are not likely to be compromised by immune memory in humans? Newcastle disease virus, vaccinia virus, herpes simplex virus, adenovirus. All right, let's see how we did. Why doesn't it show the correct answer? Well-
It's too divided.
Maybe, but no, it should show the correct answer is A. Newcastle disease virus is a chicken virus, right? Unless you work on a chicken farm, you're not going to have antibodies to it. But the others, vaccinia, herpes, adeno, they're all human viruses, so you're more likely to have immunity. Newcastle disease virus. Okay, last topic is viral oncotherapy, using viruses to destroy tumors. This is another thing, if you want to work on viruses and you want a clinical application, this is very cool. I think this is going to be a solution for many cancers in the next, 10, 15 years. So here in general, we have some non-human viruses that selectively replicate in human tumors for whatever reason. Myxoma virus, Seneca Valley virus, or we modify human viruses to target and kill tumors, and often we have immune enhancement. So I'm going to talk about both approaches today. But first, how did we get the idea to use viruses to treat tumors? Well, in fact, there were some early studies, as you can see in the 40s and 50s here, where they used human viruses to treat cancers. And this is before clinical trials were standardized. So all they did was, here, for example, in 1949, they had 22 patients with Hodgkin's lymphoma, and they decided to treat these patients with hepatitis virus, okay? They didn't know which hepatitis virus was in it. They just took it from patients with hepatitis, and they injected it into these patients. And of course, 14 patients developed hepatitis. Are you surprised? And four patients had a transient response where their lymphoma regressed, and one person died. So, that's because this is a poorly designed clinical trial. If you don't even know what virus you're putting in and its pathogenicity, that's not good, right? Next one, 1952, 1954, they used West Nile virus to infect people with various advanced cancers. All right, over 100 patients, 90% or more were infected with West Nile virus. 10 patients had transient responses in the cancer, then 10 patients had mild to severe encephalitis. So you'd never do this today with wild-type West Nile virus. 1953, treatment of leukemia with Epstein-Barr virus. Again, 3 people developed mono, which is caused by Epstein-Barr virus. And finally, cervical carcinoma, treated with human adenovirus, and there was transient necrosis in 20 patients. Now, none of these experiments yielded any useful information, and none of them would be acceptable today, but they provided just some clues that maybe viruses could treat tumors. And so from there, many people started studying viruses and oncotherapy. And of course, as we entered the era of molecular biology, we're able to understand how some viruses might interfere with tumor growth. Now, it turns out that many viral genomes you can introduce mutations in many viral genomes that impair countermeasures to the antiviral defense. Cancer cells and typically have defects in interferon system, okay? And that's part of the reason they're cancer cells. And so without interferon, the viruses could selectively replicate in the tumor cell. And if you introduce these mutations into the viral genomes, the viruses will not replicate elsewhere with an intact interferon response. So for example, in adenovirus, you can put a mutation in the E1B gene, which is normally needed for repression of ISG transcription. Remember, the interferon-induced genes, it can no longer inhibit that, so the replication is limited to the tumor. For herpes simplex, a deletion in the gene called ICP34.5 it doesn't counter PKR. Remember, PKR is a potent antiviral defense protein. And if you don't have that protein, you can't replicate in normal cells, only in tumor cells that lack the interferon system. Same thing with vaccinia virus and VSV. Specific deletions you can introduce that make the viruses unable to counter interferon. So now you render the virus specific to tumors which have a bad interferon system. Right? So these were some of the early approaches to making viruses tumor specific, because you can't have them replicating everywhere, right? That's not good for the patient. Some other ways you make, more modern ways to make viruses tumor cell selective, right? We've talked about interferon. It turns out that many tumor cells display CD46, and many normal cells do not. And CD46 is the receptor for measles virus vaccine strain. And so measles virus vaccine strain has been adapted for use to treat certain kinds of tumors because of that. The herpes simplex virus glycoprotein D has been modified so that it will bind specifically to certain tumors. Same with adenovirus. You can insert domains into the binding proteins that recognize tumor antigens. You can add adapters to the adenovirus fiber that retarget the cancer cells. So these are all at the level of binding, making the binding specific for tumor cells. And then an interesting one is the last one, mutations in the ADE1A or E1B genes that prevent countering of RB or P53. So RB and P53 are inactive in many, many tumors. And so remember, adenovirus has to counter those proteins, otherwise it won't replicate. So if you take away the ability of adenovirus to counter P53 and RB, then it will only replicate in cells that are defective in those, which turn out to be tumor cells. And so here's a little explanation for that. In fact, there was an early clinical trial of a product called Onyx 015, and it was used to treat head and neck solid tumors. And so it was adenovirus, very simple, adenovirus lacking E1B. And so here's E1B. And remember, we talked about how E1B antagonizes P53, because remember, adenovirus goes into the cell and turns on DNA replication. P53 is going to sense that and kill the cell. So the E1B protein basically destroys P53 so that you cannot stop the cell from replicating. So that was the original idea behind Onyx 15, that you make the virus specific for tumors. But there are probably other reasons too. E1B has other functions, so E1B also interferes with DNA damage repair. And so if you take out E1B, the virus can't replicate in cells where the DNA damage repair mechanism is interfering with its DNA synthesis. And also, the protein interferes with this group of proteins that detect DNA damage. And if you take it out, if you take E1B out, this will be normal and the virus will not be able to replicate. So 3 reasons that make Onyx 15 specific for tumors. Unfortunately, it's never been licensed in the US, although this is an interesting proof of concept. It went through multiple clinical trials and showed some promise. To make viruses tumor specific, you can do post-entry targeting. What we've just talked about is a kind of post-entry targeting, but here are some other versions. And so it can be either positive or negative. So for example, here's a healthy cell. The virus has a promoter. Your vector has a promoter that doesn't work in healthy cells. But you've picked the promoter that works in tumor cells, so that will restrict the virus transcription and replication to tumor cells. So that's positive targeting. And then negative targeting, you incorporate a target for microRNAs in the viral genome, for microRNAs that are present in normal cells. And so in normal cells, the genome of the vector will be inactivated, but in tumor cells which do not have the microRNA, and you have to make sure it doesn't, the virus will not be inactivated and it will reproduce and kill the tumor. And this is just a northern blot on the right, looking at 2 different microRNAs, LET7A and miR124, and you can see differential expression of these microRNAs in different tissues. So it is possible to identify microRNAs with these properties. You also want to make vectors that increase lysis of the tumor cell. So once the virus gets in, specifically in the tumor cell, it increases the lysis. So you can put P53 in adenovirus. So P53 is a protein that induces apoptosis, so that will increase lysis. You can incorporate into the viral genome cell genes that are prodrug convertases. Remember, these are genes, and I'll show you an example of this, that converts a prodrug to a toxic drug, and that will help kill the cell. And then you can incorporate genes for viral proteins that allow cell spread of virus particles to shield them from antibodies. So here on the bottom is an example of a prodrug convertase. This is acyclovir, the anti-herpes virus drug. It's a prodrug. It has to be converted by the viral thymidine kinase before it is a chain terminator. And so you use the same strategy for designing vectors to kill tumors. So we call this arming with prodrug convertases, and here are two examples. TK converts ganciclovir to GTP, which will be incorporated and stop DNA replication. Cytosine deaminase converts 5-fluorocytosine to 5-FU, which also stops DNA synthesis. So they stop DNA replication of tumor cells. So the idea is you have a prodrug, you give the patient. In a normal cell, it will not be converted and activated because there is no enzyme there. But if you incorporate the enzyme, the prodrug convertase, into the vector, then in the tumor cell you have active drugs, and these drugs will spread throughout the tumor. Here's an example of an actual drug that was tested in humans. It's called TOCA-511. It is a murine leukemia virus with cytosine deaminase. And so this was used to treat brain tumors. It's given intratumorally or intravenously with 5-fluorocytosine. So you can inject right into the tumor, both the vector and the 5-FC, or put it intravenously. And it went through 3 phases. So here's the design. Here's the design. Tumor in the brain. You inject TOCA-511. You give fluorocytosine, which is converted to 5-FU. The idea would be that that kills the tumor. However, in 3 phases for treatment for glioma, it was not effective. So this hasn't gone anywhere. We also want to promote anti-tumor responses. The tumor microenvironment is immunosuppressive, so innate and adaptive responses are disrupted. So we have a couple of strategies to get around that. We can express cell surface molecules for the checkpoint inhibitor ligands, PD-L1 and CTLA4. These will normally block T-cell responses, and we can prevent that by inhibiting them with monoclonal antibodies, for example. And we modify the genomes of many oncolytic viruses to remove genes that block interferon responses, as we've said before. And we often include in the vector genes for immunostimulatory proteins like GM-CSF that will attract immune cells to the tumor and facilitate its destruction. And so here's an example of a licensed product that has many of these adaptations in it that I've told you about. Herpes virus, it's for treatment of melanoma. It's a herpes virus called telimogene laherparepvec. Why do you make something nobody can pronounce? Laherparepvec. Anyway, it has the gene for GM-CSF. So this stimulates the production of granulocytes and macrophages, which will stimulate immunity against tumor antigens. The virus genome has been deleted for 2 genes, ICP34 and US11. Those 2 deletions cause tumor-specific replication. They have deleted ICP47, which normally inhibits antigen presentation. So now there's no inhibition of antigen presentation, and the tumor cells can be recognized by cytotoxic T cells. So this went through a phase 3 for melanoma, injected intratumorally. They had 16% responses, meaning an improvement, versus 2% for just GM-CSF alone, the drug. So it was approved in 2015. It's called Imligic. I don't know the price, though. We also use myxoma virus The same virus used in Australia to kill European rabbits. This virus doesn't replicate in any non-rabbit host, but it turns out to replicate in many human cancers. And maybe this is because the cells don't have an antiviral response, or maybe because pathways have been activated that lead to transformation. So people are trying to use this virus to treat various types of human tumors. And measles virus has also been tried for some tumor treatments. They use the vaccine strain, right, which preferentially replicates in tumors. Tumors bear the receptor CD46, and this vaccine also cannot antagonize innate responses mediated by STAT1 and MDA5. They also put in the gene for a human sodium iodide symporter. So iodine can be taken up into the tumor cell so you could see if it's effective. So during virotherapy, gamma-emitting isotopes are given, and you can visualize virus replication in the tumor. And those isotopes can also induce radiation poisoning. So the symporter is a channel that will take up the isotope into the cell. And that was used in some treatments of multiple myeloma successfully. In the end, the therapeutic efficacy of oncolytic viruses is a combination of the virus killing cells and also activation of the anti-tumor response, right? So here we have our cancer cell. We infect it with an oncolytic virus. A fraction of these are killed, viral oncolytic, but these cells will also release antigens that turn on various aspects of the immune system, antigen-presenting cells, CD4 and CD8 cells, NK cells, which will migrate to the tumor and start to kill the cells as well. So not just virus killing tumor, it's really the initial lysis activating immune responses. So this is called the abscopal effect. You can locally activate immune responses. So say a person with multiple tumors, if you infect one, the distal tumors can be also taken care of because of the activation of the immune response. All right, our last question forever. Which of the following statements about oncolytic viruses is incorrect? Infection by oncolytic viruses leads to destruction of tumor cells only at the site of virus inoculation. Some viruses of non-human animals can reproduce selectively in human tumor cells. Viruses with both DNA and RNA genomes can be developed as oncolytic agents. Various mutations in viral genomes that confer tumor-selective reproduction eliminate or impair viral gene products that counter host interferon defense. So which one is wrong? See how we did? Well, now we have the right answer. See, A, infection by oncolytic viruses leads to destruction of tumor cells only at the site of virus inoculation. That's not right. It also does distally because of this activation of the immune response. Everything else is correct. And finally, cancer immunotherapy using CAR T cells uses viral vectors. So many cancers become refractory to killing by T cells. And so what you can do is reactivate those T cells. You produce what's called a CAR, a chimeric antigen receptor, which consists of various signaling domains and a part of the protein that will engage the receptor on T cells. So you take T cells from the patient, you deliver the CAR gene via a lentivirus vector. It's produced in the tumor cells, so the chimeric antigen receptor. So you need to have something that's tumor specific, obviously. And then the T cells will recognize the CAR antigen that's been put in it. And this has been successful for certain kinds of cancer treatment. It was approved by the FDA in 2017 for certain blood cancers. So all of today's lecture should tell you the importance of basic research. All of these therapeutic viruses have been made possible because of fundamental advances in virology, recombinant DNA, immunology, clinical science, and we need this to continue. And chance plays a big role in science, right? Serendipity, people studying packaging sequences, and then it turns out to be important for making virus vectors. So we always need a balance between translational research and basic research. So going forward, I do hope this continues in this country. I'm very worried about it. All right, there you go. Don't forget what you've learned here. I've taught you a lot of virology, I know. You got lots of links and references where you can go to. You now know more virology than most people on Earth. So use it, right? Don't lose it. There are lots of places you can go that I direct. I have a lot of podcasts. I have my lectures, of course, which hopefully will happen every year. And these are links to some of the places where I am. And yeah, you can follow me in my car. I have viruses on my license plate. All right, thanks everybody.